This is Oenothera organensis, otherwise known as evening primrose. This unique flower is found only on the Organ Mountains in New Mexico and is pollinated primarily by hawk moths. The Organ Mountains stretch about 85 miles across southern New Mexico and is a botanically diverse mountain range that is home of upwards of 900 vascular plant species, one of which is Oenothera organensis. It is a self-incompatible flower, which means it can only accept pollen from another flower, and not itself. As you may have guessed by the name, evening primrose, the flower blooms in the evening around 7 p.m. in order to be open at night when hawk moths are out and pollinating. Hawk moths pollinate from dusk till dawn. Their proboscis, or their tongue, is usually about the same size or larger than its body. They forage for nectar using their tongues that act like a straw, and are primary pollinators for Oenothera organensis. Another unique characteristic of Oenothera organensis is that the style is very, very long. It is used to connect the stigma, found at the top of the flower, and the ovary, found at the bottom of the flower. And when a pollinator, such as a hawk moth, moves from one flower to another, it is responsible for transporting pollen to the ovaries.
So we're out here taking data on two different populations of Oenothera organensis. One population is an in situ population from New Mexico, and the other is an ex situ population grown in a greenhouse. We are collecting hypanthium length, hypanthium diameter, and sucrose percent from flowers from each population to compare and contrast the survivability and conservation availability of plants in the greenhouse.
As Darwin mentioned, we are comparing the greenhouse variation that was hand-pollinated to the naturally occurring species to compare the morphology and phenology of the two variations. To measure hypanthium length, we used a ruler and measured to the nearest millimeter. Hypanthium length matters when comparing ex situ and in situ populations because if it's too long, the hawk moth cannot reach the nectar. If it's too short, pollination might not occur because the hawk moth may not be able to contact the anthers, leaving no productive reasons for a pollinator to visit. Next, we measured hypanthium diameter, or flare diameter, which is the center hole in the flower. Using a pair of calipers, we measured to the nearest millimeter. While taking our measurements, we noticed a slight difference between the greenhouse and the native species. We then shamelessly sliced and diced the flower open in order to expose the puddle of nectar at the bottom of the flower. We collected samples of the nectar using microcapillary tubes. Then after we placed the nectar samples on a device called a refractometer. With this contraption you can place the nectar on the panel and look into the light to get a clear reading. And then it tells you what the sucrose percent is for the nectar. Although our results may look like there is not much of a significant difference, simple differences in hypanthium length and floral tube diameter can directly affect hawk moth pollination. As for our phenology, we discovered the New Mexico species bloomed earlier and that both variations had followed similar blooming patterns throughout the season. While taking our observations, we found a whole lot of interesting insects, such as this one, the white-lined sphinx caterpillar, which we found munching on the bulbs of our flowers. This next guy is the spotted cucumber beetle, which we found inside the floral tubes almost daily. This is a primrose moth, who adapted almost perfectly to its environment, blending into the flower to make it look like it's one of its own petals. Every so often, we were visited by some grasshoppers. Most frequently, we found the Mompha moth larvae found inside of the bulbs of flowers. These little rascals inhibited the opening of the petals, making it extra hard to measure floral diameter, and also left behind some icky looking goo.